It's my pleasure to open this session. My name is Eric Bush, Vice President of Biology for Arrowhead Pharmaceuticals. Arrowhead Pharmaceuticals is a clinical stage biopharma company based with research operations based here in Madison, Wisconsin. We are a very exciting and growing company here. Our business is the innovation of discovering and developing new medicines that utilize the power of RNA interference to selectively silence disease genes and bring new drugs to patients at need. Arrowhead is an important and growing component of our state's biohealth infrastructure, and we're very pleased to support this session, and I'm very interested in hearing today's speakers. And with that, I'll turn it over to Mike Harrison, Business Development for BioForward, to get us started. Thank you, everybody.
Hello, everyone. So this is the first annual Biohealth Communications Competition. I'm really happy that this all came together, and thank you to all the participants. There's two reasons that we started this competition this year. One is we want students to be as involved as possible, and we like to have students attend the summit, and we try and provide them as many opportunities to meet with our industry as possible. And this is another way for us to really emphasize and show that biomedical research across the state is really the backbone of our industry, and we want to highlight and support our young researchers as much as we possibly can. The second reason that we did this is, as has kind of been touched on already earlier today, public trust in science has kind of taken a hit as of late, and I really think it's important that our young researchers learn the valuable skill of being able to communicate with the public, because getting out there and being able to talk about your work and the importance of that work to everybody's lives is a critical skill, and one that's becoming more critical as the days go by. With that being said, I did want to make a couple final notes here. One is when the speakers come up, if you guys want, there are some waters here behind the podium. We're going to keep having speakers come up. But first we're going to start with a couple videos. There are two of our speakers that were unable to join us today, and so we're just going to show the videos that they sent in as an application for the competition. I will note that we had many people send in videos, and the people that you're going to see today are the 10 finalists. So these people are already winners. These are the ones that the judges selected as the 10 top videos that we saw. So without further ado, the first talk will be from Devesh Kumar, who's a medical student in the Department of Ophthalmology and Visual Sciences at the Medical College of Wisconsin.
Hello, everyone. My name is Devesh Kumar, and today I'm going to be sharing my research interests with you all. Diabetes is a chronic disease and affects many of our loved ones. And long-standing diabetes presents with eye problems such as diabetic macular edema, which is the topic of discussion today. So let's go ahead and get started. Long-term diabetes causes thickening of the back of the eye in a small neural tissue called the retina, which causes swelling and vision loss, and this is referred to as DME, as in diabetic macular edema. DME affects 3.8% of individuals over the age of 40 in the US, and it affects 23 million people worldwide. One thing to note is not every person with diabetes will develop diabetic macular edema. That is why patient screening and imaging is so important. DME is diagnosed via a high-end technique called OCT, which allows us to see where the thickening is occurring, whether there's fluid buildup in the retina, and if there's any vision abnormalities. And although the mechanism of DME is complicated, the basis behind why it happens is due to vascular endothelial growth factor, VEGF, which promotes the growth of new blood vessels. And the back of the eye is highly protected with a barrier which limits what can actually enter the retina. And because of the excess of VEGF, the barrier breaks down and there's a fluid buildup in the retina, which is what compromises the vision. The main goal of treatment for DME is to maintain good vision. And you guys all must have seen this chart before, and 2020 and 2025 are considered good vision, and anything above that is considered visual impairment. So the current research shows that someone with DME that has good vision, we shouldn't administer any treatment, but they should be monitored. And if there is a visual problem, the options include either giving them an anti-VEGF treatment to prevent the blood vessels from growing abnormally, or giving a laser treatment, which essentially burns the retinal area where the leakage is occurring to prevent the fluid from spreading, kind of like cauterizing a wound. And neither of these treatments actually restore the vision lost. Additionally, the anti-VEGF treatments are shown to be not as effective. If the patients are given 3 anti-VEGF injections without much improvement, they're actually considered non-responders. Additionally, these treatments are injected into the eye, which is quite painful. So patients often don't want to come back for their second or their third injection, but this is the only treatment that we have right now. There is a need for new treatments with different targets and mechanisms, especially ones that don't relate to VEGF. And something to really think about is, why do only a few people respond to the anti-VEGF treatments and the others don't? There must be something else going on, so we need more research. We also need to find better delivery methods of these treatments, such as eye drops, to eliminate any barrier to patient treatment, such as the painful injection. The purpose of this presentation was to really educate you all about the clinical challenges that exist with DME treatment, and I hope that this sparks conversations about what we can do to really advance the field. Thank you so much.
Yeah, you can clap. Great. So our next speaker.
Hello, everyone. My name
geez.
is Devesh Kumar, and today
All right.
I'm going
I
to
don't
be
know what I'm
sharing
doing
my
here. Okay. All right. Our next speaker is Ryan Adam, who's a postdoctoral fellow in the Department of Physiology at the Medical College of Wisconsin.
Hello, my name is Ryan J. Adam. I'm from the Medical College of Wisconsin in the laboratory of Professor Allison Kriegel. Thank you for the opportunity to present. 15% of US adults have chronic kidney disease, or CKD. We model CKD in our lab using rats. Here's a histological cross-section of healthy kidney rat tissue. Here's one from one with kidney disease. You can tell they're very different. I'd like to draw your attention to these vesicles that fill the lumens of these kidney tubules in our diseased rats. Let's take a closer look. These circular shapes have not been described before. These are not in the medical literature, they're not in the textbooks. But this begs the question, what are they? They appear to be a form of, or a type of extracellular vesicle, or EV. Now there are 3 broad categories of EVs: apoptotic bodies, microvesicles, and exosomes. Are the EVs we see any of these? Well, exosomes by definition range in size from 40 to 100 nanometers, and microvesicles from 0.1 to 1 micron. We did a size analysis of our EVs, and they averaged 2.8 microns in diameter. So just on the basis of size, we know they're not microvesicles, we know they're not exosomes. What about apoptotic bodies? Well, our EVs lacked markers specific to apoptotic bodies, such as tunnel staining and caspase 3 staining. What we have are none of these. Okay? We believe we have discovered a new type of EV. We named them large renal tubule extracellular vesicles, LRTEVs. We see them in the kidneys of diseased animals as well as in their urine. We hypothesized that LRTEVs contain kidney proteins. What is a protein? Well, in physiology, proteins are biomolecules that perform a wide variety of functions for the body. Indeed, we did find that LRTVs contain many kidney proteins. For example, I'll just highlight a couple. We found aquaporin 1. Aquaporin is important for concentrating urine. This is a key function of the kidney. I'm just highlighting one, but there's many more. Here's 8 or 9. But overall, we found hundreds of proteins packed within these LRTVs. In conclusion, in chronic kidney disease, essential tubular proteins are integrated into LRTVs and expelled into the urine. LRTVs are a new type of extracellular vesicle. These have not been described before. LRTVs may represent a never before appreciated pathogenic mechanism of chronic kidney disease. Finally, therapeutics that prevent LRTV formation may improve outcomes in people with chronic kidney disease. I'm presenting this PowerPoint, but this was a team effort led by Professor Allison Kriegel. I'd like to thank the Department of Physiology and the Cardiovascular Center at the Medical College of Wisconsin. Thank you.
Okay, our next speaker is Erin Seigan. She's a graduate student in the Department of Microbiology and Immunology at the Medical College of Wisconsin.
Thank you, Mike. So my research focuses on assessing how the immune system responds to cancer and how we can manipulate or exploit the immune system to enhance a patient's ability to eradicate a tumor. My long-term hope is to design a cancer immunotherapy utilizing a patient's own cells. In an ideal world, a cancer immunotherapy would eliminate all the cancer cells and have zero toxicity to the patient. Unfortunately, we do not live in an ideal world, but with my research, I hope to contribute to helping patients with cancer achieve long-lasting remission. So in order to develop a cell-based therapy for cancer treatment, my research focuses on 3 major steps. So that is to discover the targets present in a patient's tumor, validate and characterize whether a person's immune system can recognize those targets, and then translate these findings into a therapy by engineering a patient's immune cells to better eradicate their tumor. So currently, one of the questions I'm trying to answer with my research is what makes a good target? In other words, what about the cancer cell should we focus our attention on? And this target is what the immune system would essentially set its sight on in order to find its way to the tumor. And so I'm also interested in answering why people have differences in how their immune system responds to cancer. So if you and I have the same type of cancer, why does my immune system respond differently than your immune system to that same type of cancer? And then based on that, are there ways that we can engineer the immune system to target a cancer across multiple patients? In this way, we would not have to start from scratch every single time to design a therapy for a patient. In order to answer these questions, I take a patient's immune cells and search for those cells that are best able to clear a tumor. I then take that information from that cell and generate other cells that share the quality of that cell with the anti-tumor function. And so far in my research, I have started to screen for conserved or shared cancer mutations across patients and have started to evaluate how their immune system responds in different ways. We then utilize this information to prioritize those cells that we would want to engineer to treat the patient's cancer. And so a big component of the lab is to also utilize approaches that take the unique genetic factors of different ethnic groups into consideration when performing our experiments. Overall, our approaches in developing a cancer immunotherapy have the potential to benefit patients with cancer globally. Thank you.
Our next speaker is Michelle Roberts. She is a graduate student in the Department of Physiology at the Medical College of Wisconsin.
Okay, hi everyone at Biohealth. As Mike said, I'm Michelle Roberts. So today I want to tell you that we found markers on patient blood DNA for different types of blood pressures, but when it's measured for whole days, 24/7, instead of a single point in time at the doctor's office. A lot of us think of getting our blood pressure done only at the doctor's office once or twice a year, which is probably not enough, or some people don't get it done every couple years. But we also have different variabilities in our blood pressure that we need to keep an eye on. So for this, we can monitor blood pressures for 24 hours a day. You can do this at home or in a research clinic setting. So by blood biomarkers, I mean DNA markers that can turn the genes on and off. And they do this through interactions between lifestyle, environment, and the DNA. It's actually called DNA methylation. So why is this important? Well, high blood pressure or hypertension also leads to a number of other diseases like stroke, chronic kidney disease, heart disease. Heart disease and stroke are amongst some of the top 10 killers in our nation, and hypertension is a big risk factor. So another reason why this is important, our blood pressures are not the same throughout the day and night. we want to regulate your blood pressures and the fluctuations around the clock. We have different blood pressures types and definitions that can help us see what's going on in the body. Like our systolic blood pressure could tell us how our heart's doing. Maybe the blood pressure is high and it's putting a lot of force on our heart, making one wall of our heart really thick or hypertrophied. So we want to prevent these diseases before they even start. So we found these blood DNA biomarkers that let us know what type of issue someone might have, because we want to treat all patients on an individual level with a simple blood sample. And you might not even need to use the 24-hour blood monitor overnight and disrupting your sleep. So our study also involved African Americans, which are a underrepresented group, and they also have an increased risk of hypertension and all of the disorders that can come from that. And our main contributions to public health is that we found these blood DNA markers so that we can better help you, but we also created a new method during this process. We found these markers, but we needed to validate them or target them. So we made a targeted deep regional sequencing method that can capture these DNA methylation biomarkers. And this is suitable for clinical testing and in population screenings. And we can do up to hundreds or thousands of patients in a single sequencing run. And that could save a lot of time and money. And overall, the early detection of blood pressure problems or issues can prevent the heart disease, stroke, kidney disease, or possibly death. And this can be with a new diagnosis or some adjustments with current medications to adjust for that day to nighttime variation. So thanks a lot for your invitation and for listening.
Speaker is Wasif Osmani, who is a graduate student in the Department of Physiology at the Medical College of Wisconsin. Yep.
Thank you, Dr. Harrison. Hi, my name is Wasif Osmani, and I'm an MD PhD student at the Medical College of Wisconsin. I work in the Hodges lab where I study SUDEP. Who's that? It's a really good question. So SUDEP stands for Sudden Unexpected Death in Epilepsy Patients. Now, millions of people around the world suffer from epilepsy. 1 out of 3 of them actually suffer from uncontrollable epilepsy. And this just means that the current medications we have to treat seizures just aren't as effective in these patients. And it's actually these patients that are at a much higher risk for SUDEP. Sucks! Yeah, it really does suck. And what makes this condition even worse is that it just tends to affect young men who are otherwise pretty healthy. And the scariest thing about this is that we just don't have a good way of predicting when it's going to happen. Basically, a young person has a seizure, and then their breathing stops and their heart stops beating. It's absolutely terrifying. And so, fortunately, my team and I are working on trying to understand why this is happening so we can prevent it from happening to more people. Do that? That's a really good question, and it's actually really cool. So we're using a new animal model, not like that mimics what we see in humans with epilepsy to see what steps lead up to SUDEP. And so first, I actually give rats seizures. No, not like that though. I actually use sound waves to give my rats seizures. And then I study their brains at different time points to see what's going wrong at the very cellular level. And it's really cool. We've been able to identify molecules that we believe activate specific pathways in the brain that can cause someone's breathing to stop and their heart to stop beating. And we think that this is what actually leads to SUDEP.
You just blew my mind.
Right? It's truly mind-blowing, and actually that's not all. We're planning on testing new drugs that aren't currently being used for seizure medications, to try and stop these pathways from being activated. And so it's my goal to use this animal model to try and prevent SUDEP from happening, and then taking what we learn and applying it to humans. Also, I hope by testing out these new different drugs that affect seizures, to try and come up with new treatment options to help those who suffer from uncontrollable epilepsy.
So cool.
I think so too. I think it's really cool. And so for those of you who may not have heard of SUDEP before, thank you for taking the time to listen and to learn. My team and I are working tirelessly around the clock to find answers for those families who may have lost a loved one to SUDEP. And as a future physician, it is my hope and my goal to use these results and findings and apply them to my future patients and their families so that they don't have to experience something like SUDEP. Thank you.
Our next speaker is Edna Chang, who is a graduate student in the Department of Bacteriology at UW-Mass.
All right. Hi, everyone. My name is Edna Chang, and I am a microbiology PhD student at the University of Wisconsin-Madison. My research starts in your own backyard. It can help us address biomedical issues related to diet and nutrition, and even help us explore the galaxy. All of this is achievable by studying the very humble ground squirrel. During winter, when food is scarce, these squirrels enter a sleep-like state where they don't eat or drink anything for half the year. And this is called hibernation. In science fiction, hibernation is what allows humans to go on long-duration space flights. But the reality is, we're not close yet to getting humans to hibernate. If you were to hibernate for a few months, you would wake up with severe muscle degradation along with many other health issues. But a squirrel, on the other hand, doesn't have this problem. So what I want to know is how these squirrels are able to maintain their muscle mass during hibernation. And more specifically, I am interested in learning how the squirrel's gut microbes respond to hibernation and contribute to the squirrel's health. My labs and I found that the gut microbes help the squirrel maintain muscle mass by recycling the nutrient nitrogen. In mammals, nitrogen is usually excreted in urine as a waste product called urea. But urea can be recycled by the gut microbes into a form of nitrogen that is reusable by both the microbes and the squirrel. To study this, my labs and I gave labeled urea to lab squirrels, and this allowed us to track where in the squirrel's body the nitrogen from the urea ultimately ended. We found that the longer the squirrel had been hibernating, the more nitrogen was incorporated into their muscle protein. This was highest in late winter, when the squirrel was hibernating for at least 4 months, compared to summer or early winter when the squirrel had hibernated for just 1 month. And this showed that urea nitrogen recycling increases over hibernation. We also found the first direct evidence that gut microbes are necessary for nitrogen recycling in a hibernating mammal. Because when we depleted the gut microbes by using antibiotics, we saw very little nitrogen recycled. Our research takes us one step closer to leveraging hibernation and gut microbes as tools to improve human health issues related to diet and nutrition. It also brings us closer to the dream of longer and safer spaceflight. So, the next time you see a ground squirrel, remember that they hold solutions to improving human health both on Earth and in space. Thank you.
Next up is Michael Kindra, who is a graduate student in the Department of Chemical and Biological Engineering at UW-Mass.
Gotcha. Found the on button. We're okay. Does that work? There you go. Thanks. Cool. Okay. Thanks, guys. Okay. Let's get started. We gotta get the clicker here. Okay, can you hear me okay? All right, hi everyone. My name is Mike Jindra. I'm a chemical engineer. Chemical engineering basically means that we take low value raw materials and we convert them into higher value products. Now traditionally, chemical engineers have been known to do this by upgrading crude oil at processes that go greater than 1,000 degrees Fahrenheit. However, biochemical engineers, such as myself, prefer to use sugar and bacteria, which are happy to grow at a mild 100 degrees Fahrenheit. But why bacteria? The reason is that bacteria can solve problems that other technologies simply cannot solve. And a great example of this is the mass production of insulin in E. coli. Before this technology, the main way to get insulin for diabetics was to harvest the pancreas from 1,500 pigs to get enough insulin for a single dose for a single patient. But now we have bacteria. My goal is to push the frontier of how bacteria can benefit society by engineering them to make functional replacements for palm oil. Now, palm oil is found in nearly half of all household products and is produced at about 70 million metric tons every year. However, the harvesting of this crop has been linked to human rights abuses, biodiversity loss, and rampant greenhouse gas emissions. Now, sustainable alternatives do exist in the market. However, they are typically more expensive. And we have seen that products that are more expensive push away mainstream consumers because these consumers are unlikely to pay for them if they don't see an added benefit in product performance. This leads us to our research question. Can we engineer bacteria to make functional palm oil substitutes that are superior in function? Now, we know that enzymes inside the cell are responsible for all of the reactions going on inside of bacteria, including the reactions responsible for making these oil products. We also know that we can engineer these enzymes fairly quickly because bacteria are relatively simple organisms, and we can engineer these enzymes really over a weekend. Now, the bottleneck actually exists in testing the effects of those changes. This is a little bit harder, and this process can take days to weeks to see if a change to an enzyme relays some kind of improvement in oil production. However, due to a collaboration with researchers at the University of Illinois, we developed a process where we could improve our testing throughput over 100-fold. This technique, which is shown here, involves shooting a laser beam at a petri dish of colony to instantly determine if it's producing oils and what kinds of oils it's producing. We implemented this technology to screen thousands of different enzymes, and we finally landed on a bacteria that was able to produce up to 90% of medium-length oils in its product distribution. Now, medium-length oils are valuable and important because they have function that longer-chain oils do not have, and they are not common to vegetable oil sources such as palm oil. So in conclusion, I hope that this brings us one step closer to delivering a product that consumers can choose from that is more sustainable and also is not any kind of sacrifice in performance. And thank you a lot for listening and for having me. Thanks a lot.
Next up is Singyi Lee, who is a graduate student in the Department of Biophysics at the Medical College of Wisconsin.
Good morning, everyone. My name is Singyi Lee. Imagine you're driving home, then all of a sudden, bam, an accident happens. You realize that your neck hurts and you cannot feel or move your fingers, arms, and legs. You're brought to the ER and diagnosed with spinal cord injury. The first thing you'd ask is, how bad is that? Can I walk ever again? Unfortunately, the current diagnostic tool cannot tell you that exactly. The current method is to examine your sensory and motor functions, such as, can you feel poking here, or can you move your arm, etc. But this test doesn't necessarily predict how much you would recover. Often, MRI, magnetic resonance imaging, MRI, is used to visualize how bad injury you have in the spinal cord. The current clinical MRI can show you where the injury is and approximately how much damage you have. But this still doesn't answer the question, how bad this would be in the long term. And this is what my research is trying to answer. We want to improve the way to evaluate how bad a spinal cord injury is and how far a patient can recover. So how do we do that? We implement a specialized MRI for spinal cords to image what we cannot see with a conventional MRI. A rat model is used to mimic human spinal cord injury. Comparing to the conventional MRI, guess where the injury is? You can easily spot where the injuries are in the red area. What's great about this MRI is that it can specifically show the damages in the nerves that are responsible for your sensory function and muscle movement. And we can quantify what we see on these images. The more neurons are healthy, the better the rats can walk. Not only we want to predict how much one can recover after spinal cord injury, we also believe that our technique can reveal the potential targets of therapy. Using this non-invasive advanced MRI, we can monitor the rats with spinal cord injury if a drug or a therapy can help them walk better. Because it is non-invasive, our imaging tool can be directly translated into human. And ultimately, our goal is to be able to answer to the initial question, will you be able to walk again? Thank you for listening.
Next up is Daniel Webb, who is a graduate student in the Department of Chemistry and Biochemistry at UW-Milwaukee.
All right. Hi, everyone. I'm Daniel. I'm a grad student at UW-Milwaukee. I'm going to be talking about our work to develop an oral treatment for asthma and my work to develop an in-depth structure-activity relationship for our drug candidate. So first up, let's talk a little bit about a lead compound. So when a company or institution wants to develop a new treatment, they'll usually have one drug that's working really, really well in their initial test to treat whatever they're trying to treat, and that would be their lead compound. So for us, we have a lead compound that we call MIDD-0301. MIDD stands for the Milwaukee Institute of Drug Discovery, and this is what it looks like in its physical form, where it's pretty much just a white powder. And so this is our main compound that we're looking at for asthma treatment. Now, if we were able to take a closer look at MIDD-0301, we could see the skeleton structure here. And this is where things get pretty interesting. Here, we have a bromine on the structure, but it really doesn't have to be a bromine there. We could change that. We could put a chlorine there, or a fluorine, or something totally different. Same thing is true for this fluorine here. We could put something totally different there, or even just take it out altogether. And that's where we get into doing an SAR study, or a structure-activity relationship. And basically what a structure-activity relationship is you make as many molecules as you can that are the exact same as your lead compound, except you change one thing. And then out of all of those molecules that you make with the one change, you figure out what change gives you the best result in your test. Then you piece together all of those best changes into one molecule to, in essence, give you a super drug with all of the best things. So that's exactly what I'm doing. I'm focusing on this position here, which is called a methyl group, meaning there's one carbon there with 3 hydrogens attached, and the triangle there just means that that's pointed upwards. And so I'm working on making as many molecules as I can that are the exact same as MIDD-0301, but we're changing that methyl group there to various other different groups. And so because I'm making a bunch of molecules that are so similar to one another, I was recently asked if this research ever gets boring. But the answer to that is not only does this research not get boring, but it's the complete opposite. The really, really cool thing about the research I'm doing is all of the molecules that I'm making are completely brand new. No one has ever made them before. And why that's super interesting is because every molecule I've made has behaved slightly different from one another. Either when I'm making the molecule or analyzing it, everything's just slightly different. And it makes it a very big challenge in the research I'm doing, but it makes this research incredibly interesting. And so with that, I'd like to say thank you for listening to my presentation.
Okay, and our last presentation is Lishu Ha, who is a graduate student in the Department of Pharmacology and Toxicology at the Medical College of Wisconsin.
Is it working?
Yep.
Not the best at technology right here. All right. Thank you so much for giving me the opportunity to present. Get excited because we're about to end this thing. So if you imagine our DNA as a text, an instruction manual that teaches us how to make a human body, like any text, you can add punctuations or mark up the text in different ways, and each helps us understand information differently. For example, and this is unfortunately a real-life experience, a simple addition of a comma in my text or family group chat can make the difference between a wonderful meal together and an awkward conversation at the dining table. But this is also the theoretical underpinning of my studies. I study, in the context of pancreatic cancer, the addition of little modifications onto the subunits of DNA known as histones. So the purple balls right there. Specifically, lending from our analogy just now, I study a group of protein known as histone methyltransferases that are essentially members of a molecular editorial team adding punctuations or marking up our DNA text. They're not changing the exact letter-by-letter content of what was already written, but they're changing the way that text is interpreted, or if you're just skipping a section of the text altogether. Now, as for just skip right there. And all of that eventually affected gene expression, which, analogy again, is our molecular editorial team finishing processing the text and publishing the final article, the protein. Now, as for pancreatic cancer, to this day, despite all the research that has come before us, it is still technical difficulties, sorry about that. It is still incredibly lethal with a barely 10% 5-year survival rate with surgery. And because of the complex molecular makeup of the disease, so far few therapeutic breakthroughs have happened in the last 30 years compared to other cancers. So our challenge and my mission is to develop novel therapies. I'm unwrapping how the addition of the little modifications we just mentioned over there might contribute to the start and progression of pancreatic cancer, and how we can harness that knowledge to create new therapeutics. So far, I am using drug inhibitors of some of the molecular workers we just talked about, aka methyltransferases, and see whether that could help us curb pancreatic cancer cell growth. And I was successful. I found a few targets that mess up the cancer cell's ability to repair damages coming from various sources over here that normal cells could normally take care of, because the drug treatments messed up the methyltransferases, the molecular workers, and that in turn triggered a change in expression of proteins that are very disease significant. So I was able to find a very small link in this triadic relationship over here. Anyway, so far, for now, my field has only begun to gain popularity, and it is guaranteed to keep generating headlines and gathering the public's interest. And all of our very own ability to fill the gaps of understanding between human health and diseases really gives me great hope that my study, if continuing and successful, can help us identify the next generation of therapeutic strategies for difficult to treat diseases such as pancreatic cancer. Thank you so much.
Thank you, Alicia. You handled our weird curveballs beautifully. We threw those in just to test you. You passed. Well, so thank you all. Those were great presentations. Admittedly, I gave the students very little time to pull presentations together. They have very busy lives, and so for them to create these amazing presentations that I think are really fantastic and really show the wide breadth of research that's going on around the state deserves a round of applause. So all of these videos were viewed by our judges. Our judges are board members of BioForward from various backgrounds and fields. And although, like I said, these are all finalists, we do have some prize winners. So we have. How we broke it down is we have a first place, a second place, and a third place, and then the rest of the 10 finalists will all be receiving a $150 check from BioForward. So, Eric, if you want to get up, please, when I call your name, just step on up and get your envelope from Eric, and everyone can give you a short, brief burst of applause. So the first two names that I'll read are two presenters, that weren't able to come today. So you can give some applause to both Ryan Adam and Devesh Kumar. Okay, so our other fourth place finalists are Aaron Seigan, I did not put these in order, so Eric has to flip through them rapidly. Let's see, Li Shu Ha. Wasif Asmani. Michelle Roberts. And Singyi Lee. So that leaves our three finalists. In third place, we have Daniel Webb. In second place, Michael Jindra. And first place, Edna Chang. So again, thank you all. Judging was very, very tight. They've all produced some very wonderful and very diverse presentations. So it's kind of a difficult task to judge them. I'd hoped to have a little bit of time at the end to kind of talk to some of our winners, but we've run out of time, and I don't want to commit the cardinal sin of making everyone late to lunch. So I will just ask a couple really quick questions of all the finalists. How many of you are planning on are going into the healthcare field as a career? All right, we got a couple of them. How many of you are looking to continue on research in academia? Is that your career? Oh, got another one. All right. And how many of you are looking to enter into the biohealth industry? Got a few more. All right, great. Well, as has already been mentioned before, a lot of our in the previous presentations, a lot of our companies have tons and tons of job openings open. So we do have a career fair at the end of the day here. So please stick around and take a look. You're all excellent candidates. So another thing that I wanted to say here at the end is. So we will be having about 15 minutes to make your way down to the lunchroom. Lunch will be, there's 2 buffet lines for everyone to go into, and then just find your seat wherever you can. Fill up the dining room, try and get closer to the stage if you can. We will have our awards ceremony, I think at 12:30. And the awards are going to some amazing speakers, so it'll be a really good presentation to check out. So don't miss that. And if I could have all of the finalists stick around, we're going to take a picture of you all. But other than that, thank you for coming. And another quick round of applause for everybody, and then we'll see you later.